Hello, everybody. My name is Teresa Vasconcelos. I'm Brazilian. I'm work at the network MBNet, but my lab is in a city in Brazil, in the state of Rio de Janeiro. The name is Petrópolis. And there I coordinate a bioinformatics lab there at the National Laboratory for Scientific Computing. And today I will tell a little bit what we have done the last 2 years. I have to close this. Yes. So at my institute, we have a wet lab. You can see here the point. Yes. So we have a wet lab, we have sequence lab there, but we also have we are a supercomputer center in Brazil, the biggest supercomputer center in Latin America dedicated to scientific work. So we have the Santos Dumont, this is the name of the supercomputer, with the high performance power for analyzing parallel high process computing and also AI. And at the lab, we sequence and we work with many different types of methodology, as such as some things Eric have already talked. We sequence genomes, transcriptomes, exomes, metagenomics, and also microRNAs. And to do that, we use the high performance computing, big data analysis, and also AI methodology. And always we try to do our work relate of human health or animal and plant health, biodiversity, because Brazil will have a high biodiversity, and also thinking about biotechnology too. Oop. Okay. So as I don't have many time to explain many of the work, I summarize here the main part. We divide our lab in main part that we are working the last 5 years. So during the pandemic, of course, we made the virus surveillance, mainly concentrated in the SARS-CoV-2, but we also have all other viruses that are important to our country, such as chikungunya, Maiaro, Puxo, and Zika virus, and most of them are transmitted also by mosquitoes. But we also like to understand the relationship between the virus and the host, the human. So for that reason, we sequence also the patient that had some disease related to Zika or dengue or chikungunya to try to understand which genes, which resistance some patient has when they have the infection caused by one of those virus. And related to COVID, we have studied a lot along COVID. So we work with precision medicine. I will talk about more about that because we are sequencing 3,000 genomes, 30,000 genomes in Brazil. We also work in the concept of One Health, as Eric explained, mainly related to antibiotic resistance, but also we are interested in collaborate with other groups to understand, to sequence the animal and plant health, and also monitor work related to the biodiversity, monitoring the ecosystems, and in the way to protection and the restoration, and also biotechnology to identify biomarkers or molecular targets. And I will show fast two works made by my doctoral student, my PhD student. One was related to metagenomics, and then for that we have sequenced samples from cows, poultry, human, and coal from different parts of Brazil. As you know, Brazil is a big country, and in this first study, we decide to understand the resistance present in many different regions of Brazil, antibiotic resistance. And we have sequenced a lot of samples. By the end, you can see here, most of the genome that we're able to assemble using one methodology that we call MAGS, I don't have time to explain this, but we still have many genomes that we are not able to achieve main species less than that, or genus or phylum. So we studied, we identified the resistance present, antibiotic resistance present in many different parts in Brazil, but a new one, we were able to identify new resistance that came Brazil from the frontiers or other way. But we will still have many results that can be used for another studies. And this, for that reason, Douglas, this guy here, up, this guy here, we decided to study the sporulation, the probability of sporulation in some MAGs that we have been identifying. Sporulation is a natural mechanism from bacteria that can be dormant in our organism or our soil, mainly some of them related to the pathogenicity. And then, but we are not able to identify really these genomes in the sample. So we develop a classifier using machine learning. We are training with the reference sequence. We divide our training in two data sets using the sporulation and non-sporulation already identified genomes. And we use different methodologies to analyze these, to extract these features. And of course, we use a stacking assembly approach, and we use different methodology, and we perform different metrics to analyze this. And at the end, we infer a novel sporulation species across these three species, cow, poultry, human, and swine. And this is the result of one of his articles. We are able to identify a novel species of bacteria present in some of the organisms, and some of them are comparted, are shared with the other species with probably with ANI, the more than 99%. In the same way, in another work with Amanda, is the Amanda thesis, we would like to see the metagenomes in sample from the mosquito, because most, as I told you, most of the virus are transmitted by mosquitoes. Most of the virus in Brazil are transmitted by some mosquitoes. And then, but there is a problem, but when sequence metagenomics, the size of the reads are so smaller that we cannot able to identify through good an alignment and assembly some genomes and extract features to classify, to identify this new virus. In this thesis, we are develop not out. We are trying to use BLAST using the CAR-MER strategy that is a well-established methodology to do that. But we try to use another alignment-free feature extraction that we call MNMER. That is a traditional variation of the CAR-MER method. And as you can see here in blue are our methodology, and then MER, and in red is the traditional methodology. But when we use this methodology with the K, the size of K ranges to 2 to 4, in the small fragments from 300, 1,000 base pair, we have better results than the CAR-MER results. We are very happy with this methodology, and then we apply in developing this workflow. The name is Morvir, and then we analyze the data sets from transcriptome in order to identify new variants in 10 minutes. Okay, in the dark matter of these samples. And all of this software are available in GitHub. You can download, you can use it and everything. So using this methodology, I have to be fast. Using this methodology, we are able to identify in this metatranscriptome, some of them generated in our lab, more than 600 mosquitoes associated that at the moment has not been described yet. Moving to another part related to precision medicine, we are also sequencing a lot of genomes and exomes related to disease predominant in Brazil. So we worked with Parkinson's disease, rare disease, immunodeficiency disease, Alzheimer, most of them high prevalence in Brazil. Yeah, and also we work with the chikungunya infection. We sequence the genome and exome the many patients that they have chikungunya disease causes inflammatory demyelinating disease that's very high, and we are able to identify some response immunity, and then can be responsible for this demyelination in the patients. And then at the lab now we are concentrate, as I told you, we are sequencing more than 30,000 genomes in Brazil related for different disease or our related our population, indigenous, and we have a miscegenation. We are very mixed population in Brazil. So we divided the country in many parts, and now we are sequencing and a lot of genomes in Brazil. And then we are hired for the Minister of Health in Brazil to develop, to help some methodology to analyze this data. And then we are start to create this platform that is a platform related to AI. And then we are going to sequence our population, and we are going to integrate the molecular data, the clinical data, but also to help to annotate, to have some studies to analyze with using scientific article these genomes. And to do that in a specialized way, secure and curated, we will use AI, and then we are able to be used for different application. This methodology, this platform will be free for our groups in Brazil, and maybe in the future for the whole community. So we're using LLM, GLM, and PLM to do that. So the first thing to do that is to have the data acquisition and the knowledge-based construction. So you use data from the literature. Only the data from the literature we will use to train our models. We always able to analyze the omics data, and the ontological data based also will be integrating this platform. The purpose is build a centralized, carryable knowledge foundation for all the models. And the second part is the RAG. This is the function to do that is a dynamic retrieves relevant information from knowledge base. And the output will be produced scientifically accurate, context aware, and reference anchor tests to mitigate the model hallucination that is happen in a lot of this case. And the third part of this platform will be is the engine for analyze the omics. And in this platform, we will analyze the genomics and the transcriptomes that will be generated for the network. And these engines will be to feed the RAG pipeline to analyze the data. And the user interface will be a chat box and also can be available using API. So this is more or less the picture of the platform. So the user can make answer, or he will receive answer that can be text or table or VCF or a list of genes. We will upload a scientific article related to this disease that we are sequencing. We will have different database separate for the scientific articles, and the people that also can submit their files or BAN or VCF that can be performed the analysis if the group that doesn't know how to do the bioinformatics analysis. We have already a pipeline to analyze all this data, and they can use our pipelines that will be compared to the genetic database available, but we install only in our server. And then we use the LLM. We are using different methodologies, different large LLM here, and also we are using genomic language, and we will start using our scientific database and etc. And then we will have a web service that can be available. In this context, this is Cynthia Rebosa, professor that's specialist in neurodegenerative disease. We start to use this platform in using these genomes, the exomes and genome that we have sequenced together. And this is very important in Brazil, but also for the world. And we start using that. So we are sequencing, so we provide for doing this study, we provide this platform. I have already explained. We are filtering periodization, interpretation of the genomics and metagenomic data. We are studying this disease, intellectual disability, ALS, Alzheimer's disease, and Parkinson's disease. And we will identify we have already identified some structural important variants in this data, and now we are to spend that for the transcriptomics analysis. And this is only to show the pipeline to analyze these variants that are able something. I identified today that some things is in Portuguese, but the name of the database is here. So is the workflow that we are analyzing in order to arrive for the variants that have high impact, moderate impact, and etc. And then that's it. I don't have time to show another work, but this, as Eric told, is important to collaborate. Our lab is celebrating this year 25 years, and this the country that we have been collaborate since during this 25 year, and we open to collaborate for more country or the same country that is here. So would like to, this is my group at the lab, and this is the two groups that we are working together to develop this AI platform. And these are the main group that support our work in Brazil. So thank you so much.